within the cell. Do we always have higher calcium outside? Or should we always have higher calcium outside? Yeah. And this is, stop taking notes for a minute. This is directly related to exam 2, parathyroid hormone and calcitonin. Do you remember them? They maintain a blood calcium level. So those two hormones you learned before that directly play a part in maintaining higher calcium levels outside so that we can contract our muscles. I'd say that's pretty important. All right? So here we go. And in these red bulbs here, what's inside these? Acetylcholine. That is our neurotransmitter. We put neurotransmitters in these synaptic vesicles. And when calcium rushes in, it creates a tidal wave movement that forces the synaptic vesicles to fuse with the synaptic ending and release the neurotransmitter into the synaptic cleft. The process of release of acetylcholine, of fusion of vesicles to the wall, is called exocytosis. You guys remember that?
So it's not a chemical or mechanical, I mean, it's not a chemical thing that happens between calcium and synaptic vessels, it's more of a mechanical
Yes.
pressure.
It's like a wave of fluid coming in with calcium, and it pushes. These synaptic vesicles are simply bouncing around in a fluid-filled container. When calcium rushes in, it pushes them toward the wall. It's the same membrane that surrounds the vesicle that is surrounding the motor neuron, and they fuse together in exocytosis. Now, this is in the synaptic cleft. It's got to be going somewhere, right? What channels might we find on the postsynaptic membrane in this case? Because this is a muscle. We'll call it the sarcolemma. What channels are here? Good. Chemically gated sodium channels. And that acetylcholine will come in and bind here, opening the channel, and sodium, which is also at a higher concentration on the outside than the inside, will rush in. And now we've transferred the voltage change from this cell to this cell. And we depolarize, create an action potential on the muscle cell, and we carry that voltage change down the T-tubule. So what type of channels would be found on the T-tubule?
Voltage-gated calcium.
Voltage-gated sodium. The chemically gated sodium channels open, and it's just like a bunch of dominoes. All right? If I set up these pins, I don't know if I can do it. I'm going to make a mess here, but if I set these up like dominoes, and this is how the channels are set up in our system, all you need to do is knock over the first one, conceivably, if these are dominoes. Y'all with me on that? And the first one gets knocked over. The first one, the trigger, is the chemically gated channel. And once it gets knocked over, the other one should open up as well. And the other ones happen to be voltage-gated sodium channels. So once you knock over the first domino here, click, 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 these are all voltage-gated sodium all the way down, and they carry the impulse all the way down the T-tubule. But we know the T-tubule ends at a very nice structure called an SR, sarcoplasmic reticulum. We're inside of a cell. And And the dominoes that were headed down the T-tubule actually will trigger the opening of the channels on the SR. You ever seen that where they set up dominoes in a gymnasium and they'll kick over the first one and all of a sudden they go out and they make all that? You guys know what I'm talking about? YouTube, like dominoes in a gymnasium. They set up 1 million of them and they're all perfect, and they click the first one and all of a sudden they make these beautiful patterns and they go all over. Same idea here. You kick the first domino over, it goes down, and this one here, when it opens, it's going to open the adjacent one, and it's going to go a different path. But what happens to do, what kind of channel is this right here? It's voltage gated calcium, because calcium is going to rush into the cell from high to low. Is a voltage gated calcium. So do you need to know where the locations of these channels are? Yeah, be helpful in the story. All right. If you want to go to my A&P place, chapter 9, look at the MP3 tutorial. There's actually an audio of this process that you can listen to if you're auditory. And it also has a 3-page narrative that explains exactly how this happens. It's what I ask you to do, write a narrative or listen to it or whatever. It's already done on A&P Place. They're called MP3 files, MP3 tutorials. Kind of explains this for you if you want to read the narrative. Good idea. Where's calcium going? Where's calcium going? Haven't heard it yet. There is one binding site inside. Where are we? We're inside a muscle cell now. You all with me with this? We're in a muscle cell. Let's go to troponin. Boom. Done. All right. That's it. We go to troponin and we did it. I sent a signal from here and all of a sudden I'm out at troponin. So calcium's coming over here, and this is where your picture has to keep going. And we're taking all of our text out of our notes and we're throwing it up here. Then you get to draw the fun stuff there. We'll do a snowman, which is our troponin. We'll do our tropomyosin. What are we going to have to put down here? Or myosin. Now connected. Calcium comes in here. Woo. Binds to there. What's that going to do? Going to open a binding site because it rotates tropomyosin off. Myosin head reaches up and does a
Power stroke.
Power stroke. Beautiful. During the power stroke we give off ADP and inorganic phosphate, correct? And you can just do the next thing. It's locked up here and now we're tied up together. And we're attached. And we need ATP to come flying in and detach. Correct? And then it drops down and we hydrolyze ATP and we recock and we're ready to go again. We got to come all the way back here. Get that to happen again, do all this stuff again, and it goes down here. All of a sudden, the only problem we got is that magic, ATP? This flow, we just say, yeah, ATP comes in. Gotta go back a little bit. Better come over here. Tell me a big, nice story about metabolism, of which the products of the 3 types of metabolism, in essence, we've talked about in here, CP, anaerobic, and aerobic, generate the ATP. So I know you know where ATP came from because it doesn't just come flying in. And if you put all this together in a nice big drawing, then you don't have to worry about your notes as much. You just have to tell me the story. And that is the story. It's hard to believe because here I am. And all of a sudden, my brain says contract. And all of this happens like that. Whenever you move Everything I just put on the board that took me 20 minutes to write out is happening. And then we clear it up. When the contraction's finished, what's the chemical that comes in here and breaks this down? Acetylcholine esterase. There's 3 things that happen to acetylcholine in the cleft. 3 things. Acetylcholine esterase will break it down. The second thing is acetylcholine, because this is a fluid-filled cavity, acetylcholine often drifts away from the receptor. No response. And the third thing is we take it back out presynaptically again. Why remake it? What's the process of pulling something from the extracellular fluid and putting it in your cell? It's called endocytosis. You already learned that. We endocytose that neurotransmitter back in. All of that. So is this your third exam? Can you explain it to me? At the level we talked about today, the textbook goes a lot deeper. The IPCD goes a lot deeper. It really is a conceptual, can you figure this out from here? Now let me check your cheat sheet. My cheat sheet. Here's the exam. I want to make sure there's no surprises. Good. Talked about the channels, talked about glycolysis, pyruvic acid. One question I didn't address, I'll just give it to you because I never addressed it ever. Why do they call it biceps, triceps, and quadriceps? Look it up. Number of origins. Good. Origins. Done. That's another easy one. Now it's easy. It wasn't easy if you didn't know that. It's the only thing we didn't talk about in here that made the exam. I think it's kind of important. You would have figured it out probably, but you may not have. Anything else? This drawing exercise is actually on Moodle. It's called Drawing Exercise for Muscle Physiology. It kind of leads you through what I'd like you to draw. Do it if you want to. It's your time. I don't know. If you're sitting there going, I got this, you're done. Don't study anymore. Go have fun. Study for other classes. All right? Now, fun. That's what, you go out, you go have a good time. That's what I mean. If you're sitting here and you have no idea what I'm talking about, you got a week. You got a week to put it together. All right?